Hello and welcome. Today we're going to explore the fascinating biochemistry behind antigen-presenting cells, or APCs. These are the immune system's messengers, the link between innate defenses and adaptive immunity. But how exactly do they form, and what molecular mechanisms allow them to present antigens to T cells? Let's break it down step by step. Step 1. Origin and differentiation. APCs arise mainly from hematopoietic stem cells in the bone marrow. Through differentiation, some progenitors become monocytes or dendritic cells, while others mature into B lymphocytes. When monocytes leave the bloodstream and migrate into tissues, they can differentiate into macrophages or dendritic cells, depending on cytokine signals such as G, M, C, SF, MCSF, and IL-4. This transformation isn't random. It's guided by transcription factors like Pu.1 and NF-kappa B, which activate genes for antigen uptake, processing, and presentation machinery. Step 2. Antigen uptake. Once an APC is formed, the next key step is antigen capture. This can happen through phagocytosis, where large particles or microbes are engulfed into phagosomes; endocytosis or pinocytosis, where soluble antigens are internalized; or via receptor-mediated uptake, such as Fc receptors binding antibody-coated pathogens. At the biochemical level, uptake triggers actin cytoskeleton remodeling, driven by Rho family GTPases. This allows the plasma membrane to wrap around the antigen, forming a vesicle. Step 3. Antigen processing. Inside the APC, the antigen must be processed into peptides. Here's the biochemistry. In macrophages and dendritic cells, engulfed antigens enter the endolysosomal pathway, where proteases such as cathepsins break them down. The resulting peptides are loaded onto MHC Class II molecules. These MHC molecules are synthesized in the rough endoplasmic reticulum and initially bound to an invariant chain, EE, which prevents premature peptide binding. In late endosomes, the invariant chain is degraded, leaving a small fragment called CLIP. With the help of HLA-DM, CLIP is exchanged for an antigenic peptide. For MHC Class I presentation, cytosolic proteins, like viral proteins, are degraded by the proteasome. Peptide fragments are transported into the ER by the TAP transporter, where they bind MHC Class I molecules. Step 4. APC maturation. When APCs detect pathogens through pattern recognition receptors like toll-like receptors, they undergo biochemical maturation. NF-kappa B and MAPK signaling pathways are activated, leading to upregulation of MHC molecules on the cell surface, expression of co-stimulatory molecules such as CD80 and CD86, and secretion of cytokines like IL-12, which shape T-cell differentiation. Step 5. Antigen presentation. Finally, the processed peptide MHC complex is transported to the cell surface. The APC is now ready to engage T cells. MHC Class I peptide complexes interact with CD8+ cytotoxic T cells. MHC Class II peptide complexes activate CD4+ helper T cells. This antigen presentation, combined with co-stimulation and cytokine release, initiates a precise adaptive immune response. So in summary, APC formation is a multi-step biochemical process: stem cell differentiation, antigen uptake, peptide processing, MHC loading, and presentation. Without this molecular choreography, our immune system would be blind to pathogens. Thank you for listening.